Welcome everyone. Welcome to the second of the Myco-Talks in our series of monthly Myco-Talks. My name is Gordon Brown and I'm based at the University of Exeter. My co-chair today is Sarah Gaffin, who's based at the University of Pittsburgh. So we have two great talks for you today. The first talk will be by Dr. Liz Ballou, who's a Wellcome Trust Sir Henry Dale Fellow, who's based at the University of Birmingham, who's going to be telling us about Cryptococcus Titan cells. And then our second speaker is Professor Bruce Klein, who's based at the University of Wisconsin. He's going to talk to us about the impact of genetic variation at the IL-6 locus on susceptibility to fungal infections. So if you haven't joined us before, just to point out that the purpose of this webinar is to really provide a platform for the community, for you, which is open access, in which we can all share in the amazing diversity of research that contributes to the broader field of medical mycology. And so the idea is that there will be two talks every month, and we're going to try and have those talks to reflect a variety of different aspects of the field, and also different aspects of career stage, etc. Now, each talk will be 30 minutes, and we're not going to have questions in between talks. There'll be the first talk followed directly by the second talk. And then after those two talks, we will have the Q&A session. Now, I'll point out, if you'll see at the bottom of your screen, you have a Q&A and a chat. Please only submit your questions to the Q&A, because that's the only part that's going to be monitored. And what will happen at the end of both talks is that Sarah, my co-chair, and I will then alternate between our questions between each of the speakers. And we will get through as many questions as we can in the remaining half an hour that we have after the two talks. As you probably are aware, we record these talks and they can be found on our YouTube channel, and you can see the web address of that YouTube channel at the bottom of the screen. And that channel also will give you an indication of upcoming talks in the future. And I've shown you some of the talks for the next 2 months that are listed there. One just important point to highlight is that you must register for each session. You will not be able to access the talk unless you do that. And you should get notification from the team about upcoming sessions, but you can also refer to the website for those talks that are coming in the future. Okay, so before I hand over to Liz, our first speaker, I just want to say thank you very much to the team behind us all who do the fantastic job in getting this to work so smoothly. And that's Jules, Lorna, and Alberta, and it's really their hard work that makes this possible. Okay, so that's me finished. I'll stop sharing my screen now, and I will hand you over to Liz.
Hello, let me just get myself set up. Hold on 1 second. All right, can everyone see my screen? I hope.
Yep, Liz, you could.
Okay, great, thank you so much. Sorry, it's not started at the beginning of the talk. Oh goodness, I'm sorry about this. Okay, so I wanted to start by thanking the organizers for inviting me to speak today. It's really a pleasure to be able to speak to this audience. But I also just want to say this is probably one of the most intimidating talks I've given, and I apologize if I'm a little bit rushing in this. So part of the reason it's so intimidating is because when you're giving a talk like this, the first thing you should consider is the audience. Right? Who is in the audience, and who will be, what will people gain from your talk? And since I don't know who's in the audience, and I don't know who will be in the audience, I decided to give this talk to direct it at myself about 5 years ago. Someone who's coming up in the community, someone who's trying to solve some challenges of becoming a researcher. How do you make that transition? And how do you think about your research in the context of a larger problem? And so I'm particularly speaking in the room to people who are looking for ways to join the community. And I particularly want to emphasize that because this is a story that has really benefited from the mycology community and from interaction with other members in the group. So with that, I will just start. The title of the talk that I'm going to give today is Cryptococcus neoformans, yeast-to-Titan transition is inducible and regulated morphotype. And this is the organism that we're going to be talking about today. Just a few things to notice about this organism. It is a very uniform organism. It's a round budding yeast. It produces by budding and division. So you can see here's the mother cells, here's the daughter cells. And they have this very regulated process, right? I just wanted to take a few seconds to talk about why this is important. There is definitely a continuing need for research into fungal pathogens. And I like to start all my talks with this wider context of the research, particularly how important it is to think about fungal infections in the context of other infectious diseases. This is a graph that I've made looking at the number of malaria deaths, tuberculosis deaths, and HIV/AIDS-related deaths this is up until about 2015, these numbers reflect. What I hope you can see is that these are diseases that are still affecting a large number of people every year, but comparable numbers of deaths are resulting from fungal infections, and this is a major neglected area of research. One of the things that is particularly striking about this graph when you start thinking about the number of deaths versus the percent mortality, which is shown in yellow, is that although there are still large numbers of people dying from malaria, tuberculosis, and HIV, the percent mortality, the likelihood that you will die if you get these diseases is really actually quite low, and this represents a success in research up until this point. However, when you look at a comparable measure for fungal diseases, and this goes right from the big three all the way down to the more neglected fungal diseases, what you see is there is a very high level of mortality with these organisms. So if you become infected, you have a much higher chance of dying. And this is a big research need in the community. And so if you're a young researcher coming up and you're thinking, is there a place for me in this field? I hope that this graph conveys that yes, there absolutely is a place for you and there's a need for you in this field. One of the big challenges that has been part of the sort of fungal narrative is that it's concomitant with HIV. So as HIV infections have increased over time, the prevalence of people with fungal infections has also increased. And progress in treating deaths in HIV has really plateaued. And this is due to a lack of attention paid to the underlying diseases that are killing people who have full-blown AIDS. And when you break that down, it's about 15% of those deaths are due specifically to the organism that we're going to be talking about today, which is Cryptococcus, with another 15% due to tuberculosis. So really comparable numbers. We estimate that there are 1.5 million fungal infections every year and 5.5 billion pounds spent on treatment. So this is a major, major area with need for progress. All right, so let's get back to Cryptococcus neoformans. This is what you're looking at is a movie that I made as part of my PhD work, looking at the way that we study Cryptococcus for many years. It's a budding yeast. This is the way that when people were studying it, they were looking for specific things. So one of the major sort of dogmatic pieces of advice you get when you start with research with a fungal pathogen is that you should make sure your cells are happy and growing actively when you start your experiment. So they need to be in log phase, they need to be well fed and actively growing. And this is what that culture looks like, right? You've got your yeast producing daughter cells in a very regulated way. And there's a couple striking things that I want you to notice about this. The first is the uniformity. The cells are very round, right? You get a very regular cell shape. And this is something that's striking to people who first look at Cryptococcus for the first time. And the other thing is that the daughter cells, they're all budding basically at the same time, right? And the daughter cells that are being produced are pretty much the same size as the mother, so proportional daughter size. And this is the way we've been studying this for many years, and because of this, we have a very well understood cell cycle. So you go from having one content of your genome to two contents of your genome, the daughter cell emerges, the nucleus is divided across the septum, and then you end up with a daughter cell that's about the same size as the mother and has equal DNA content, right? And these cells are about 5 microns in size. Okay, so the problem is that when you look in patient samples, that's not what you see, right? Instead, you see very, very large cells and very heterogeneous cell populations. So this is an abscess from a young Zimbabwean woman from a study that was done in 1973. And when the young woman came in, they took a lung sample, and aspirated this pus from her lung and put it on a slide, and this is what they saw. They have these very, very irregular population of large cells. This is a 40 micron scale bar. So you can see there's, these guys got up to 100 microns in the sample that they described. You have a number of small cells as well. So this is really a heterogeneous population that is quite distinct from what we see in the lab. You can see that same sort of situation in a brain section from one of the first patients that was diagnosed with HIV/AIDS back in 1983. This is a young American man. And again, what you're noticing is that, and rather than this very uniform population, we have quite a lot of heterogeneity, right? We have small cells, we have large cells. The staining of the cells is quite distinct, so that there's different intensities in fungal cell wall. So what you've got here is a conflict between what we're seeing in the lab and what we're seeing in patients. And this was sort of noted in the literature, but not a lot was followed up with until in 2001, Marta Feldmeiser in Arturo Casadevall's lab tried to replicate this. So she took some cells, a variety of different strains. Specifically here, we're showing pictures of serotype D, B3501, and the ATCC strain 24067. And she took these into mice, and she looked at what the cell sizes were in mice. And what she noticed was what had been a very uniform population in the lab now became this highly heterogeneous population in the mice. And you can see here some very different cell sizes. This is a 10 micron scale bar just for reference. But we've kind of pretty much left it there as a field. So despite this observation that in mice, yeast can take on this giant cell morphology, it wasn't really until 2010 that this became reproducibly understood to be a true phenotype, a true in vivo phenotype. And this is work that was led by two different labs, Kirsten Nielsen's lab, so Laura Okagaki and Kirsten Nielsen's lab, the graduate student working on it at the time, and Oscar Zaragoza in Arturo Casadevall, a postdoc who have been working on this for many years. Both of these labs back to back were able to show that when you take H99, so this is the same strain that I showed you at the very beginning of the talk, and you put it into mice, what you get again is this very heterogeneous population. So you can see the very small fungal cells here inside of an abscess, but as well as these very large cells, which they've dubbed Titan cells. Oscar Zaragoza took these large cells and then grew them on rich media. And what you can see is, in contrast to the regular division that you see in a YPD culture, you instead see this asymmetric division and a production of a very small daughter cell. And this is a very reproducible part of this phenotype. Okay, so this is pretty interesting because now we've got this new in vivo phenotype. And subsequent work from Kirsten Nielsen's lab has demonstrated that these cells have a particular number of particular features. So they're defined as being anything greater than 10 microns in diameter, and this is cell body diameter, so we're not including capsule diameter here. They have this thickened cell wall that you can see quite dramatically in this figure that Laura took. They have altered capsule, which I'm not going to discuss in this talk, and they're also highly polyploid. So where instead of having just one genome content, they have 4 times their genome content. We think they're quite important because they're associated with reduced phagocytosis, and you can see, for example, here macrophages being entirely overwhelmed by the physical size of this Titan cell, whereas normal fungal cryptococcal cells is represented here in blue. They're associated with a non-protective Th2 response, more rapid dissemination to the brain, and poor outcome in patients. But despite all that work, we had a big challenge because what we were looking at is an in vivo phenomenon, but we lacked an in vitro model. And this is the big question that I came to when I started my own lab, is how do we bridge this gap between having a very reproducible and important in vivo model to actually working in vitro model. I'm not going to go into a huge amount of detail about what I did to sort of solve this problem, but I'm very happy to talk about it if people have questions in the question session or just want to reach out afterwards to have a conversation about this. But basically what we were able to do was, based on a little bit of hypothesis and also, frankly, quite a lot of luck, we were able to, in my lab, solve this problem. And I will just take you through that story a little bit. So the model in the assay that we finally developed, we grow cells in minimal media. We put them at a low density. So we typically use 0.001 as our density into PBS with 10% serum, put them at 37 degrees and 5% CO2, and that's to mimic the host lung. After 24 hours, what we see are a typical yeast with their nice fluffy capsule, but also these very, very large Titan cells. This process of how we came to this see this was I've described in a paper that I published this year in MSphere, and you can, if you want to read about sort of the story there, I'm very happy. There's a reference there, and I'm also happy to discuss it after the talk. But what I want to highlight is that we weren't the only people to discover this. Instead, it was actually 3 labs back to back to back who all independently came up with different methodologies to grow cells in this way. So here's our methodology, and this is summarized in a review that my PhD student in my lab and I published a couple years ago. Along with our assay, Oscar Zaragoza and Alexandria Lanio's lab also came up with 2 different methodologies for growing these cells and inducing these Titan cells. And this is a sort of unusual thing in the field where you have competing labs coming up with different methodologies for studying the same phenomenon, right? What was really wonderful was that through open conversation and through sort of protected collaboration, the 3 of us, the 3 labs, were able to publish the stories back to back to back in PLOS Pathogens. And again, I've described that process in the MSphere article that I wrote. And what I think is really important is, number one, the fact that we have these 3 back-to-back papers shows that the phenomenon is extremely reproducible. And different papers have found different things, so we each contributed something unique to the story. But also there are some commonalities across this. So for example, minimal media is quite important. Starvation media conditions is quite important. Cell density was shown to be quite important, which I'm going to talk about a little bit later. Yeah, so here's the story about cell density. So one of the things that was really interesting is that cell density is a major determinant of cell size. This is a figure from our paper where I show that if you grow cells in different concentrations, so high concentration, this is 1 OD, 0.5 OD, 0.025 OD, going down and down and down, and which I hope you can see quite dramatically is that over the course of this dilution, you can get larger and larger cells and a more and more heterogeneous population. And Nuria Treviano-Contador nicely showed that this is, in the Zaragoza lab paper, nicely was able to show this is due to the effect of the quorum sensing protein. So dilution of the quorum sensing protein results in these very large cells. So there's a sort of a correlation between those 2 papers. All right, so now that we've got this nice in vitro model and we've got multiple different ways of growing these cells in vitro, what can we actually learn? Well, one of the most striking features for me was that the Titan cultures are highly heterogeneous. And I think you can see that if you just watch the difference between the way that a Titan cell culture grows and the way that, comparing to previously, the way a yeast cell culture grows. So in a Titan cell culture, you get this very large cell that's produced here with a large vacuole in the middle, and it's producing daughter cells that are small, right? They're disproportionately small compared to the mother cell. And what you end up with at the end of that is a culture that looks like this. It's got large Titan cells, it's got typical yeast cells, and it's got these very, very tiny Titanide cells, which we're calling Titanide cells. And so we tried to understand that a little bit more. One thing we did was to ask, well, what happens if you take Titan cells and you culture them again? So if you take the small cells that come out of a Titan culture and you grow them in the context of Titan-inducing conditions again, what you can see is the production of new Titan cells. So here we're taking a Titan cell that was from an old culture, calcifluor white was added, and then the cells were allowed to proliferate. And what you can see is this cell is no longer staining, indicating that it came into the culture after the calcifluor white was added. But it's a true Titan cell with capsule similar to the normal Titans of the Titan cells of the previous culture. These are heterogeneous populations, and actually there are 3 cell types present. So you can see in these, if you do TEM, you can see typical yeast cells, which have a kind of a typical cell wall. You can see the very, very large Titan cells, which have a much thicker cell wall. So these are all to scale here. And you can see a 3rd cell type, which we're calling Titanides after the sisters of the Titans. And you can see here they have a distinct oval shape. They have a nucleus, and they have a very, very thin cell wall that is just significantly thinner than either yeast or Titan cells. So in addition to this large Titan cell, we have this new cell type called Titanides. We think these are really interesting cells, and we have a paper that is just describing these. It's currently on bioarchives if you're curious to read them. But I think one of the most important features of these is that these Titanides really do seem to be emerging from Titan mothers. This is work that was done by a postdoc, Masha Makarova, where she took our Titan culture And she stained it with a ratiometric dye that reports lipid order. And because lipid order is heritable, the daughter cells of will have a similar lipid order to the mother cells. And if you just take that analysis, what you can see is that Titan cells have a particular order, a high order to their cell membrane compared to yeast cells, which are less ordered. Some very small daughter cells have that same order compared to the mother cells, which is graphed here. And here's just a video showing a Titan cell. These are all Titan cells here, producing these small daughter cells. So hopefully we can convince you now that Titanides do arise from Titan mothers, at least in part. And if you want to read more about what we think those things are, what's interesting about those cells, you can find that on the bioRxiv paper. We can also, by using this model, learn about regulation of what's the things that are driving Titanization. So we were able to reproduce work that had been done in vivo, showing that the cyclic AMP pathway is quite important. So these are all genes that are involved in cyclic AMP regulation. And if you knock out any of them, you're able to block the ability of cells to become Titan. So remember cells that are Titan are anything that's greater than 10 microns. We were also able to find negative regulators. This is a transcription factor that when you lose it, you get a much higher number of Titan cells. We are also able to investigate the question of whether capsule is required for Titanization. And here's a Cap59 mutant that's not able to make capsule, but still is able to make cells that are greater than 10 microns, suggesting that there's a separation between the production of capsule and the production of Titan cells. Okay, so this process that my student discovered, Tom, was that if you take cells and put them at low concentration and then put them in the presence of serum, then you're able to get Titan cells. So this is a two-step process. And so we wanted to look into that a little bit more. What we found was that if you take serum and you fractionate it, and then you ask what portion of the serum is actually associated with Titanization. So there's a nice clean peak here. You can see these guys are Titanizing. Cells induced with this portion of the peak are not Titanizing. And if you take that peak and you start doing deeper and deeper analysis of it, eventually what you'll get is an MR spectra that looks like this. And this spectra is consistent with a compound called peptidoglycan, which is a component of the bacterial cell wall. I'm showing you the structure of peptidoglycan here. It's a really interesting molecule. Even though I'm a fungal biologist, I can acknowledge that bacteria are interesting. And this bacterial molecule, peptidoglycan, is basically part of the bacterial cell wall. It's quite distinct from fungi in that it has a particular sugar backbone, which is glycnac and then murnac, so N-acetylglucosamine and N-acetylmuramic acid. And then it's cross-linked by this peptide backbone that links these sugar molecules together. And that's distinct from the molecule that makes up this fungal cell wall, chitin, which is glycnac, right? So this portion here is going to be unique to bacteria, whereas this portion here is going to be more conserved to fungi. If you just take this portion here and you add it to cells that have been primed to form Titan cells, so that's this muranyl dipeptide, what you can see is that that is sufficient by itself to trigger the increase in cell size that we see. Likewise, you're able to induce Titan cells if you grow them in the presence of E. coli, heat-killed E. coli, so they don't need to be alive, or strep pneumoniae. And we can also reproduce the phenotype if we just take bronchoalveolar lavage from a mouse lung and use that. So we think this is really important because it's demonstrating that this is a cross-kingdom signal, right? Bacteria are providing a signal that's causing this change in fungal morphogenesis. The last concept that I want to talk about is the change in genome content that we see in these Titan cells, because I think this is a really fascinating concept, but I just want to give a little bit of background to this concept because it is slightly complicated. So hopefully you all understand that DNA is contained in histones and it's packaged into coils, and these coils are then organized into chromosomes, right? And in diploid organisms like humans, we have 2 copies of all our chromosomes. This is actually not chromosomes, this is socks, but I think it's a cute way of showing that chromosomes in humans come in pairs. And in situations where you have aneuploidy, which is the incorrect number of chromosomes, so for example, here's Down syndrome or an individual with a trisomy of chromosome 21, now you can get this imbalance in chromosomes. We see this process in humans, right? We also see it in fungi, I just want to define a few terms here. So organisms that are haploid have 1 copy of each chromosome. Organisms that are diploid have 2 copies of each chromosome. an organism with uneven balance in chromosomes. So for example, this individual would be considered to be aneuploid, right? And an organism with an even balance of chromosomes is considered to be euploid. So that would be this person represented by the socks. We can analyze genome content using a process called flow cytometry and DNA content analysis. So that's represented here, where this is from an H99 culture where we've got a peak representing one content. So this is what we normally see in a yeast culture. Two content, again, now it's duplicated its genome, it's going through its cell cycle, and you can also see where it would have a tetraploid content. So if it was aneuploid and it was producing a further duplication of its genome, you'd have 4C, right? And if we take Titan colonies, what we can see is that shift from having a normal 1,2C content to having a 2,4C content. Okay, so one of the things that we understand about Titan cells is that they're highly polyploid. So when we look at our in vitro induced cultures, so again, here's our 1C content and our 2C content of our yeast phase cells. If we look at a Titan culture, instead of seeing this normal phase, what we instead see is this very distinct stepwise increase in DNA content. And so here you can see a nice clean peak of what the majority of the cells having a haploid content, but again, it's a highly heterogeneous population, right? So some will have 4 times, some will have 16 times, sometimes will have 32 signs, and some will have 64. You can even imagine there's another peak here that might be an even further increase. And those very, very high DNA content cells are also the ones that are extremely large, right? So there's a proportional relationship between the DNA content and the size of the cell. You can visualize that. So if you look at individual single cells, so here's a typical yeast culture. So you can see our cells are stained here for showing chitin. The green here is representing the nuclear membrane, and the red are representing individual chromosomes. So in a yeast phase culture, your nuclear membrane and your yeast phase chromosomes are here, and we can actually count the individual chromosomes in there. On average, we see about 9. It's not all the chromosomes in the genome, but we can sort of get an idea of what an average content of a haploid cell is using this methodology. And hopefully you can see that in typical yeast cells, you have nice representation of individual dots, but in a Titan cell, there's so many chromosomes here that you're no longer able to resolve individual runs. Instead, we can just take it as a fluorescence intensity to approximate that. In contrast, daughter cells that are produced by these Titan mothers, now we are able to visualize individual chromosomes again, indicative of an unequal division of DNA and a unusual sort of cell cycle that it's going through. The conclusion here is that Titans in fact produce aneuploid daughter cells. And if you actually analyze the DNA content of those daughter cells, what you can see is this atypical plot, right, showing that the daughter cell is either diploid and going through a cell cycle from 2 to 4 instead of this more typical 1 to 2. And if you analyze a whole bunch of different daughter cells coming from individual colonies, what you can see is that the majority of them, in fact, are diploid, right? If you take that and you go back after 30 days and you check, what you'll see is that some of them have reverted to haploidy, some of them are maintaining what appears to be diploidy. I've now done further experiments where I've come back after a year, so these cells went into the freezer and came back out after a year, and now they've all reverted back to haploidy again. So this is a transient process. Why is that important? One of the major concepts when you're thinking about fungal pathogens is to think about drug resistance. Drug resistance is a major, major challenge, although it's not necessarily appreciated the way that bacterial drug resistance is. And one of the major differences between Cryptococcus drug resistance and other fungal pathogen drug resistance is where drug resistance, for example, in Candida albicans is associated with point mutations in the genes that are actually the targets of the drugs. So for example, ERG11, which is a major target of fluconazole, we frequently see point mutations in ERG11 that prevent binding of the drug. Instead, we rarely see that in Cryptococcus. What we see instead is this process of genome duplication, particularly amplification of 2 chromosomes, chromosome 1 and chromosome 4. Chromosome 1 is particularly interesting, and this is work that was done by Junquan Cheng's lab, showing that chromosome 1 is particularly important for drug resistance because it contains both the ERG11 target as well as an azole transporter. And when you get amplification of that chromosome, you quickly become resistant. So just looking through that process here, here is H99 in the presence of fluconazole. You can see that fluconazole is clearing the fungus. If you wait long enough, you quickly get resistance, right? If you take those colonies and you look at their genome content, you can see aneuploidy in chromosome 1 and chromosome 4 associated with resistance to this drug. If you take the cells off fluconazole and then look at them again, you can see that there's been a loss of this drug resistance and a loss of aneuploidy at these chromosomes. So it's this transient resistance, right? What's interesting is that because titanization is a mechanism for driving this aneuploidy, we asked, well, is it actually a mechanism for generating heteroresistance? Other research from Kirsten Nielsen has shown that, yes, these aneuploid daughter cells are strongly associated with ability to resist drug, but what we've added to that is that if you grow cells under Titan-inducing conditions, just that process of growing the cells under Titan-inducing conditions is sufficient to generate resistance to fluconazole. And this, again, is probably due to the aneuploid that we're seeing in the daughter cells that come out of this process. Okay, I just want to conclude with one last thought, which is that, yes, Titan cells give you lots of frequency of drug resistance emerging, so you can definitely see the ability of these cells to resist drugs is increased. But because they're polyploid, they also face unique challenges. This is work that's building off of beautiful work that was done by Audrey Gash and published earlier this year, showing that aneuploidy in Saccharomyces cerevisiae is strongly supported by the activity of a protein called SSD1, which is involved in post-translational regulation of various different transcripts. If you look at SSD1 mutants in Cryptococcus, you don't really see a phenotype. There's not much going on there. It's only when you look in aneuploid cells that you're able to see a phenotype, and we're able to demonstrate that beautifully here looking at an SSD1 mutant under Titan-inducing conditions. And what I hope what's immediately apparent is that Titan cells have a profound defect in cytokinesis when they lose SSD1, but yeast cells have no issue, and that's because they're not aneuploid, they're euploid, right? They have a normal genome content. So in addition to Titan cells providing cells with a ability to resist stresses, for example, drug stress, as well as others described by other researchers, Titan cells present unique challenges to the fungus because they have to be able to overcome particular stresses associated with being polyploid. Okay, I'm going to stop there, but I hope what I've convinced you is that Cryptococcus exhibits phenotypic diversity both in vivo and in vitro, and that by priming for titanization, by studying cells in the context of titanization, we are able to understand heterogeneous morphologies that are important for immune evasion. We believe this is more reflective disease. It has major implications for drug resistance as well as lung persistence and latency. And when you grow your cells under these Titan-inducing conditions, rather than a uniform population, you get this very highly heterogeneous population that presents lots of opportunities for study as well as opportunities for the fungus to evade the immune system and cause disease. I need to conclude by thanking all of the people in my lab, including my graduate students, my postdocs, visitors to the lab, my collaborators on the SSD1 project, and there is a bioarchive paper on that project if you want to read it, if you're interested in learning more, focusing more on the structure of SSD1 rather than particular functions. I really owe a huge debt of gratitude to my Titan collaborators, including Ivy Dambuza, who is my immunologist, go-to immunologist, although I didn't talk about any of the work that she did today. I just want to acknowledge her, as well as other more senior researchers who've been really supportive of my career. And I just want to end on that, if I can just get one more minute from the team to just talk about sort of a philosophical thing there. I think one of the most important things that I've learned through this project is that simply changing growth conditions in a fungus, we're able to see a really fascinating and important contribution and ability of the fungus to thrive in a lot of different environments. And for example, just by growing the cells under Titan-inducing conditions here, we're able to see macrophages controlling the fungus, capsule-mediated evasion. You can see vomocytosis here with these yellow cells. There's just normal extracellular growth and this process of titanization actually occurring within a macrophage. So just by changing the conditions here, you can see a huge amount of really interesting stuff happening. And the only reason that I've been able to study that and work on that process is through the support of a network of mentors, some of who I've named throughout this talk, and who you can also see documented in the other papers that I've described. Last year, I had the pleasure of working on a paper talking about the power of mentorship with Sarah Gaffin and Neil Gao and a couple others. And so I just wanted to point people to that if you're looking for a mentor. and looking for ways to be a better mentor, we've got a section at the end of that paper that describes some resources for everyone. All right, I'm going to stop there and send you back to Gordon for the second talk. Thank you so much.
Thank you very much, Liz. That was great. Very nice talk. I'm sorry you can't hear the 200 people, 250 people we have attending applauding you, but I'm sure everyone is, and there's lots of comments coming in. If I can please just remind everyone that if you have questions, we've got lots of questions that have come in, but if you can, just remember to put your questions into the Q&A box down below. That would be great, and we will address those questions after Bruce's seminar. So thank you very much, Liz. Bruce, it's your turn, you're up. So Liz, could you stop sharing the screen?
Yeah, no problem.
All right, Bruce, are you good to go?
All right, can you hear me, Gordon?
I can hear you, but we're on your presentation view, not on your slide view. You had it right the time before.
How's that?
There we go, you're good.
All right. I wanted to start by pointing out that probably many of you are familiar with Where's Waldo? What I'm showing here is the New Zealand Where's Waldo edition. And I think the current social distancing edition will speak to many of you out there in the audience. So thanks to Gordon and the MRC Advisory Board for inviting me to talk about genetic variation at the IL-6 locus and human susceptibility to fungal infection. I'm going to focus on blastomycosis in the story that I'm going to tell you. Blastomycosis, or blasto, is one of the 3 North American endemic mycoses, which also include coccidioidomycosis and histoplasmosis. These infections due to blastomyces, coccidioides, and histoplasma are, as we know, primary pathogens, in contrast to crypto or candida infections, for example. The dimorphic fungi infect otherwise healthy people and account for probably close to 1 million new infections every year in North America. They're very common causes of pneumonia in this area. There are a number of Mendelian genetic predispositions. For example, deficiencies in interferon gamma, IL-12, or IL-17, but these are pretty rare in the population. In contrast to that, we know there are racial and ethnic variabilities in disease susceptibility or severity of disease that have been described in Filipinos, African Americans, Hispanics, Native Americans, and then most recently in the Hmong population in Wisconsin. And these variabilities are thought to be the result of polymorphisms or SNPs in the population, but none have been described to date. This is one of the holy grails, I would say, in the field of study of endemic mycoses. What accounts for the variable susceptibility among these different populations? And I'm going to talk to you about what we discovered in the Hmong population in Wisconsin. So blastomyces, as I mentioned, is a dimorphic fungus. It grows in the environment, typically in the soil, in the mold form. And the organism and infection can be found in North America, east of the Mississippi, in many of the states in the US. also up into Canada, and then also in a number of countries on the African continent. After the organism is inhaled into the lungs of a susceptible host, it undergoes this quite striking morphologic transition to the pathogenic yeast form at elevated body temperature of the mammalian host. And this is the cause of pneumonia, which often disseminates outside the lung in about half of the cases. The first responders to blastomyces are typically neutrophils and macrophages, and Th17 cells are very important, as we've shown from my lab, in adaptive immunity. And collectively, these elements of innate and adaptive immunity constrain infection and account for subclinical illness in about half of the cases. So just as sure as the snow flies in Wisconsin and other regions of the endemic area, there are outbreaks pretty much every year or two. And while exposed individuals have asymptomatic infections in about half of the cases, we also know that about the other half develop progressive pulmonary blastomycosis. We investigated a large outbreak of about 60 cases in central Wisconsin in 2009 and 2010. And one of the most striking features of this outbreak was the predilection of infection in the Hmong population, which was heavily overrepresented among the cases. The CDC and the Division of Health investigated the cases going back over the previous 5 years, and we found that the rates of infection in the Hmong population were sometimes 10 times higher than they were in the European ancestry population in Wisconsin. So the big question here is, why are the Hmong so susceptible to blastomycosis? And we hypothesized, not surprisingly, that Hmong individuals may have genetic variations that put them at risk, at increased risk for developing blastomycosis. So to define, or to begin to define, some of the candidate susceptibility variants, we hypothesized that they were probably common in the Hmong population, rare in the general population, and they would meet 3 biological plausibility criteria that I'll tell you about in a moment. And so we sequenced the whole genome of 11 Hmong patients from the outbreak, and we performed homozygosity mapping, looking for runs of homozygosity in which both alleles were identical across all the patients' genomes that we sequenced, and that these were rare in the general population, comprising less than 20% in Europeans, and that these alleles contained either a regulatory feature or were associated with gene expression, for example, harboring EQTLs or expression, quantitative trait loci, or that they harbored a known antifungal immunity gene. So in all, among these runs of homozygosity, we identified 113 candidate variants that distinguished the Hmong from European ancestry individuals. Most of the runs of homozygosity that we identified harbored anywhere from 1 to 5 candidate variants. However, one run of homozygosity was really remarkably distinct. At the IL-6 locus, there were 24 variants that stuttered the entire locus. So returning back to Th17 responses that are important in controlling blastomycosis, what we know is that the differentiation of naive T cells into Th17 cells, Th17 memory cells, is heavily dependent on interleukin-6 as a nurturing signal in conjunction with IL-23, TGF-beta, and IL-1. So we hypothesized that the Hmong individuals may have genetic variants that alter the production of IL-6 and consequently impact the Th17 response in comparison to the general population. And we were agnostic about whether the Hmong might have too little or too much IL-6. You could imagine that either of these could be deleterious, but simply that they lacked just the right amount of IL-6, the so-called Goldilocks hypothesis, if you will. So we tested IL-6 production by immortalized cells from European ancestry individuals and from Hmong patients that were part of this outbreak. And we looked at B lymphoblastoid cells, or LCL cells, that were immortalized because we were concerned that we would have trouble getting Hmong patients back for repeated visits, given their concern and superstition about mainstream medicine. We stimulated the cells from Europeans and Hmong subjects, and then 24 hours later, we harvested the supernatant to measure IL-6 and, as a control stimulus, IL-8. IL-6 levels in response to a stimulus were actually significantly lower in the Hmong patient cells than they were in European ancestry subjects. And in surprising contrast, IL-8 levels as a control in response to a stimulus were actually significantly higher in the Hmong as compared to the European ancestry subjects. So the Hmong actually have reduced IL-6 responses in comparison to otherwise healthy European ancestry subjects. So because IL-17 is so important, IL-6 is so important in the development of CD4 positive IL-17 memory cells, we looked in the peripheral blood of these individuals. And we isolated CD45 RO positive memory CD4 positive T cells from the blood of these individuals in conjunction with CD14 positive monocytes as antigen presenting cells. And we stimulated these cultures with Candida albicans. We chose Candida albicans because we know that most individuals, healthy individuals and others in the population, are colonized with Candida albicans and have developed immunity. In contrast, most individuals in the endemic area probably have not been exposed to blastomyces. And we harvested the supernatant of these cultures about 4 to 5 days later. What we found was that the IL-17 producing CD4 positive T cells in response to Candida were actually significantly lower in the Hmong compared to the European ancestry individuals. And similarly, ROR gamma T expression, which is the sort of sine qua non transcription factor for IL-17 producing cells, was also significantly lower in the Hmong population compared to the European ancestry controls. So IL-17 production by antifungal memory cells is actually reduced, and this defect could conceivably even extend beyond blastomycosis to other fungal infectious diseases. So we also directly tested, in a complementary approach, the role of IL-6 in susceptibility to blasto in our animal model. And here we vaccinated with a live attenuated strain, wild type and IL-6 knockout mice. We boosted, challenged, and analyzed them several weeks later. And we also had unvaccinated control animals. What we found was that the CFU levels in vaccinated animals was actually significantly higher in the IL-6 knockout group compared to the wild type controls. And likewise, the IL-17 producing CD4 positive memory cells was actually lower in the IL-6 knockout group compared to the wild type group in the vaccinated cohort. And likewise, IL-17 production by stimulated splenocytes was also significantly lower in the IL-6 knockout group compared to the wild type group in the vaccinated cohort. Conversely, we didn't see any differences in unvaccinated animals when comparing the wild type group to the wild type to the IL-6 knockout group in the unvaccinated cohort. So IL-6 is essential for the development of adaptive immunity, particularly Th17 immunity in the setting of experimental blastomycosis, as well as showing associations in the human condition. So then the question is, how do the IL-6 variants explain susceptibility in the Hmong population? So for this, we turn to an analysis of the 7th human chromosome that harbors the IL-6 locus. And what I'm showing here is the sense strand of IL-6 in red with exons in boxes, and the anti-sense strand in blue, which actually is a long non-coding RNA or a link RNA. And what I'm showing here in the vertical lines are some of the variants that we identified, with those in red actually being predicted expression quantitative trait loci, or EQTLs. So for example, this one at position -572 being in the long non-coding RNA and also known to be an EQTL. So it turns out that antisense IL-6, the link RNA, is an enhancer of IL-6 gene expression, an enhancer. And so candidate variants may impact not only antisense IL-6, but conceivably also the IL-6 product. So for example, it's known that the EQTL at position -572 within the link RNA is associated with anti-IL-6 expression, which is actually highest in Europeans that have GG at this -572 position, and lowest in Hmong individuals that are homozygous for CC at the -572 position. Admittedly, there are only a small number of Hmong that have been tested to date. More than that, it turns out that in silico modeling predicts an impact of these candidate variants and on antisense IL-6. So at the EQTL in position -572, we know that the CC allele that's so common in the Hmong predicts an elongated, highly rigid RNA structure, in contrast to the prediction for European ancestry individuals that have a homozygous GG allele, where the RNA structure is predicted to be much more flexible and nimble. And this is thought that conceivably it could influence RNA stability or microRNA binding, for example. So to test these predictions, we turn to gene editing in human stem cells, and we isolated induced pluripotent stem cells from a Hmong donor and from a European donor, and we then edited the genome of these iPSCs using CRISPR-Cas9 technology. So as an example, I'll focus on position -572 in the link RNA, where we know that Hmong are homozygous for CC in nearly 98% of the population, which is quite rare in the European population, comprising less than 5%. And conversely, Europeans are homozygous for GG at position -572 in over 80% of the population, which is exceedingly rare and hasn't been found in any of the Hmong that have been studied so far. So in the iPSCs, in the European parent line, we edited the GG sequence at position 572 to a CC that's typically found in the Hmong population. And conversely, in the Hmong iPSC parental line, we edited the CC sequence to a GG sequence commonly found in the Europeans at position -572. So if our hypothesis is correct, we would predict that the parental European line edited from GG to CC at this position, 572, would actually reduce interleukin-6 levels. And conversely, we would predict that the Hmong parental line that's edited from the homozygous CC sequence to a GG sequence at position -572 would actually result in elevated levels of IL-6. So I probably wouldn't be showing these data if they didn't turn out to be interesting. So what we found in the European cell lines is that the IL-6 levels in response to a strong stimulus of IL-1 beta and zymosan were high in the parental line, and they actually fell quite significantly when we edited the GG to a CC that's found typically in the Hmong at position 572. And likewise, in the Hmong parental line, we found that the IL-6 levels in the stimulated cells exposed to zymosan and IL-1 beta were low in the parental line, and they rose significantly when we edited the homozygous CC to a GG typically found in Europeans at position -572. Now, I'd also quickly point out that you can notice that the IL-6 levels in the Hmong parental cell line are once again quite a bit lower than what we found in the European line, where we see nanogram quantities of IL-6. So while position 572 certainly has an effect, it's likely not the only polymorphism that explains the altered production in the Hmong population. Nevertheless, there's no doubt that position -572 has a significant impact on altering cytokine production, IL-6 production at the Hmong locus. So to conclude this story and perhaps catch us up a little bit in time, our study unveiled a novel genetic underpinning for the elevated risk of blastomycosis that's been linked to Wisconsin Hmong ethnicity. The antisense IL-6, which is a long non-coding RNA or link RNA, regulates IL-6 and has a polymorphism that varies and contributes significantly to the differences in IL-6 production between European ancestry individuals and Hmong individuals. And I think that our findings could forecast a broader vulnerability to fungal or other infectious diseases in patients that are being treated with inhibitors that block interleukin-6, sort of reminiscent to TNF alpha inhibitors. And I should quickly add that we're now using quite widely IL-6 inhibitors in the clinic, including in the setting of COVID-19 infections. And finally, IL-6 is quite a pleiotropic cytokine whose regulation is not completely understood. And link RNA, antisense IL-6, regulates expression of IL-6, but its mode of action requires quite a bit more investigation. So I'll end by acknowledging the folks that did most of the work that I showed you. Richard Merkhofer was an MD PhD student that just graduated from the lab and is back in the clinic. Mary O'Neill was a PhD student in genetics, working in Caitlin Pepperell's lab, a close collaborator. And there were a number of other contributors to this story from my lab and from Caitlin Pepperell's lab. So I'll be happy to stop there and let people in the audience ask questions of both the speakers, and turn the program back over to Gordon and to Sarah. And thank you once again for the chance to tell our story.
Thank you very much, Bruce. And again, there's 200 hands clapping, more than 200 hands clapping for you in the background, but this is the one bad thing about this forum is we cannot share that with you live. Jules, can you open my video, please? It says that you need to allow me to open it, and also that of Liz's, please. Okay, there we go. Thank you. Thanks very much. So thank you, Liz and Bruce. Thank you very much for those wonderful talks. Very clear. There's been lots of enthusiasm for it in the comments. And I'm going to start off with a question for you, Liz, which is coming from several people about the mechanism of sensing of the peptidoglycan that induces this Titan. Do you have any ideas how that's happening?
Yeah, this is a really important question that we've been addressing in my lab for the last probably 18 months now. I don't have a complete story to tell you about this yet, but some really interesting preliminary data that I didn't think was quite ready for this talk. Basically, what's happening is that peptidoglycan is it's this unique molecule that's only found in bacteria. It's something that's sensed as a calling card. I think one of the questions was, how did peptidoglycan get into serum? Peptidoglycan is basically everywhere. Anywhere that there's not a sterile environment, you're going to get peptidoglycan. It's sensed by mammals, it's sensed by plants as a major sort of calling card of the presence of bacteria. It seems like fungi can also sense it. I've shown it. Also, it's been seen in Candida albicans to trigger hyphal growth. And so one of the major questions is, how is that actually happening? And that's what the work that we're doing right now. And I think I'll stop there and just ask if there's, maybe I'll wait for the next question to give you more information.
Okay, thanks, Liz. Hand over to Sarah.
Sure, Bruce, that was great, really enjoyed that. There are several questions that sort of tended to the same issue here, which is having this polymorphism potentially selected for, and as such, is it seen across other populations other than just the Hmong or even this particular family?
Yeah, so I'm sure it's being selected for. As we know, too much of a good thing is not very good. And there's probably some advantage in terms of the origins of the Hmong population from tempering down elements of IL-6 supported immunity. So while I don't know specifically the selection mechanism, I would surmise that there has been selection for tempering down Th17 elements of immunity for where these individuals have arisen. You know, we do know that this probably extends beyond the Hmong into a broader Asian population. There are variants, and they certainly do affect susceptibility to different disorders. IL-6 variation and variants at IL-6 are associated with difference in susceptibility to including disorders including cancer and especially tuberculosis. So yes, it's probably more prevalent.
Right, Liz, my one. So again, there's been some interest about the impact or the development of Titan cells and whether you see it in other cryptococcus species and whether there's something similar perhaps in other fungi.
Yeah, okay, so two things there. One, we definitely see Titan cells in both Cryptococcus neoformans, the serotype A, as well as serotype D, the day neoformans isolate. And that's been demonstrated both by my group and as well as in the other two papers that were published back to back. There was just a really nice paper that came out from Lucas Kozłowski's group that looked sort of more broadly at other relatives of cryptococcal species. And using our assay, they were not able to see Titan cells in any other isolates outside of that direct lineage of the pathogenic neoformans. I suspect that it's probably because the inducing conditions are not quite the right conditions to induce these, or possibly that our definition of what a Titan cell is not going to apply uniformly across the species lineage, right? Does that make sense? Yeah. The question of whether they've been seen in other fungi, I think this is a really fascinating one, and it really comes down to what is a Titan cell, because we're defining it based on size, morphology, DNA content. But if you're thinking about it more as a developmental change in response to peptidoglycan, then it actually has probably already been shown to happen a very similar phenomenon in Candida albicans, right, by Yue Wang's lab. So whether there will be similar changes in response to peptidoglycan in other organisms, I think is a really fascinating question.
Okay, there was a question about that's to deal with this with about Paracoccyloides brasiliensis, whether they've seen Titan cells.
Yeah, I mean, I don't know if the word Titan cell is the right thing to call them, but I think it'd be fascinating to look.
Okay, thanks.
Bruce, so there was a question about, you've looked at the circulating Th17 cells. There's a question about the tissue resident Th17. I'm willing to guess you may not have that from the humans, but maybe you have some insights from the mouse studies.
Yeah, so you're talking about natural Th17 cells?
Well, the question is comparing circulating memory Th17 compared to tissue resident.
Right. So we haven't looked at tissue resident cells. I'm wondering, so the answer is no, not in this particular model. In other models, we have looked very carefully at tissue resident memory cells, particularly in our vaccine model, in animals that get recombinant endoglucanase, which is a very potent vaccine immunogen. And surprisingly, we found that when we elicit large numbers of tissue resident memory cells, including IL-17 producing cells, by administration of vaccination at the mucosa intranasally, those cells don't function in a way that we would have expected in mediating vaccine-induced resistance or adaptive immunity, in contrast to eliciting cells systemically that reside in the spleen or in the vascular tree within the lung that hustle to the lung upon challenge. And they instead are the ones that are important in mediating adaptive immunity to a challenge. So sort of unexpected in terms of tissue resident cells that mediate adaptive immunity, at least in our vaccine model.
Yeah, some reminiscent of the TB vaccine too.
So there's two questions that have come in that I've come across so far that talk about the sensing of peptidoglycan by crypto in the lung, and whether that's possibly an evolutionary outcome of it dealing with bacteria in the environment. And related to that question is whether you see Titanization only in certain anatomical locations in the lung.
Yeah, okay, so crypto isn't an obligate pathogen, right? It's not something that's always in the lung or in human lungs or in animal lungs. It's something that's associated with soil, with pigeon guano, with sort of the environment, right? And as a soil-associated organism, we would expect it to frequently come in contact with bacteria, right? So the framework that I'm using to think about this is that this is just a normal part of the Cryptococcus life cycle that it does in response to environmental stimuli. And in fact, growth on YPD or growth on SABDEX, the way we do it in the lab, is probably not the normal way that these cells are growing in the environment, right? If we think about the human lung as a structure, one of the major concepts people have been considering for many years now is that yeast cells are pretty large, right? 5 microns is pretty large. And there's a barrier in the lung that prevents large particles from penetrating into the alveoli. this is why the work that Joe Heitman's been doing and others, Christina Hull and others, has been on the fungal spore as an infectious propule has been so important, because the spores are very, very small, they're oval, they can get down into the alveolar space. What's interesting about the Titanide cells that we've identified is they're very similar in shape and structure to spores, and so they could potentially be part of that dissemination process. This is a hypothesis that we're in the midst of testing right now. So when you think about the where Titan cells are forming, the cell has to first get to that spot, right? If we're thinking about sort of histology, we see Titan cells in mice lungs pretty much everywhere. We haven't looked at human lungs.
So is there evidence for Titan cells then in the environment?
To my knowledge, no one has ever actually directly observed Cryptococcus in soil or in the environment. People can culture it, you can recover it, but there's no evidence of what the actual niche is in terms of, what its morphology is in the environment. But this is a project that actually, if COVID hadn't happened, then we would be trying to tackle this right now. But
okay, thank you.
Bruce, there's quite a few questions about autoimmunity and this SNP. So you sort of talked a little bit about evidence for evolutionary conservation, but maybe you can speak to this, particularly Th17 driven autoimmune diseases like psoriasis, for example.
Yeah, so as you well know, Th17 is really important this gets to the issue of the Goldilocks hypothesis, perhaps. Too much of a good thing is not very good, and Th17 cells are important in autoimmunity, and there are a number of different inhibitors of various aspects of Th17 development, IL-17, in the clinic. There's certainly no doubt that IL-6 is also implicated in altered inflammation and autoimmunity. And inhibitors of IL-6 are being used in the clinic for treating these disorders. So there's no doubt that fine-tuning of the amount of IL-6 and the amount of IL-17 is critically important. And these disorders or alterations that point to the consequence of not having just the right amount are really illustrative in terms of the human condition and what they teach us, about autoimmunity and basic biology. Yeah. I would add that, as I mentioned, an IL-6 inhibitor is widely being used now in the clinic for treatment of COVID-19 infections, particularly tamping down what's believed to be the cytokine storm in the second phase of COVID-19 pneumonia and multi-organ system involvement. So there's been some benefit that's been reported in anecdotal cases from modulating IL-6 levels in that setting.
There were two questions that are sort of vaguely related, so I'm going to try and stick them together a little bit. So one question is about the clinical strain. Did you see variation or at least low levels of Titan formation in H99 coming in? And then is there increased frequency in clinical isolates? But then there was a comment also that actually the relative frequency of seeing Titan cells in clinical samples is quite low. So are you able to tie those two together in terms of the pathology and the role of Titan cells in pathology is what I'm trying to say?
Yeah, okay. So we have looked through a screen of clinical isolates to see what percentage of them form Titan cells. And this is in the Dembuza-Drake paper, if you want to look. About 30% of clinical isolates definitely form Titan cells. You can easily see them. About 20%, it's more difficult to say, is that really a Titan cell? It seems like it's changed morphology compared to its yeast phase growth, but it's not hitting that 10 micron cutoff. And about 30%, we're not able to see any evidence of Titan cells. I've got a paper on bioRxiv specifically characterizing the spectrum of those different clinical isolates in more detail in a few of them, although it hasn't quite made it past review yet. So maybe take that with a grain of salt. In terms of actually seeing Titan cells in clinical isolates in the clinic, as yet there's no evidence of people seeing these, for example, from lumbar punctures from CSF samples. And whether that's because they don't form in the CSF, so they form in the lung and then they disseminate as smaller cells and then form as Titan cells in other sites, or if that's because there's something special about the limitations of the CSF nutrient-wise, that's unclear. It is clear that if you take Cryptococcus yeast phase cells and put them directly into the brains of mice, they will form Titan cells. So it's not something specific about the lung.
Okay, thank you.
Bruce, so you alluded to the fact that the sort of a prediction of the structural changes in the RNA for IL-6 might be related to this intrinsic stability of that message. Have you actually tested that?
No, not yet. Coming to a theater near you. We're very interested in actually looking at the RNA and testing some of these in silico predictions, but we don't have any data yet. I mean, this is, you know, the role of link RNAs is really an emerging area. And a number of link RNAs have been studied in terms of their pleiotropic contributions to gene regulation. So we imagine that this link RNA might be involved not only in regulating IL-6, but conceivably, based on some predictions, could be regulating other targets outside of the IL-6 locus, acting in trans at other sites. So we're really interested in studying this link RNA, I guess suffice it to say, but we haven't yet done a deep dive.
So I have two unrelated questions that I'm going to try to put together for you,
Okay.
which are around about the formation of and formation rate of Titan cells. So the first part is if you delete the quorum sensing features of crypto, does it change the rate at which Titan cells are formed? And the second part of the question is the rate of Titan cell formation in vitro similar to what you see in vivo? Or do you know the answer to that?
Yes, so one of the tricks we do is to work with QSP1 mutants. Yes, they make more Titan cells. Still not 100%, but they definitely make more. It's more like 30% instead of 20%. The rate of Titanization in vivo, so I think the question that was asked was actually about the timing, the speed at which Titan cells first emerge. That's not my work. Kirsten has done that work and has shown that you can see them actually quite early during infection within the first couple hours. So I won't comment too much on that. But I would predict that, so what we already know is that Titanization frequency, the number of cells in a population that are Titans is much higher in vivo than it is in vitro. And we've got a paper that will be submitted in the next 6 months showing that's probably because of the additional effect of the host immune response on the process of becoming a Titan cell. So things that happen in vitro are predictive, but are not directly related to what happens in vivo.
Great, thank you.
Bruce, so there's a question, which I'm just going to read it verbatim. IL-6 can also be produced by adipocytes and inhibited by estrogen. So this person would be interested to know if blasto-infection risk correlates with body fat or gender.
So does the risk correlate with body fat or gender? That's a great question. So what I can tell you is that in the human population, men are at much greater risk than women for contracting sporadically occurring cases. This probably has more to do with the fact that men are more likely, not all men, but in general, most are more likely to be fishing, hunting, tromping through the woods, and getting exposed to the organism in the environment than are women on average. When you look at outbreaks, which really controls for some of these confounding variables, there's really no gender effect on susceptibility. And when you look in experimental animals, there doesn't seem to be a gender effect in susceptibility. So I guess I would conclude that in terms of hormonal effects, there doesn't seem to be a gender influence. Obesity, you know, that's interesting. So there is potentially an association with obesity in that individuals that are larger and suffer from diabetes, for example, as a consequence, have a significantly greater likelihood of contracting infection sporadically. And, you know, one wonders if this could stem from adipocytes and their response, or alternatively, there are certainly underlying effects on elements of immunity, for example, on neutrophil function, which are so important in restraining infection with blastomyces. So I guess, you know, from what we're learning about the influence of weight and adipocytes on the immune system, I would say that there's tantalizing evidence that there may be an effect. Yes.
So I have questions around drug resistance, Liz. So we have one question that relates to whether fluconazole resistance correlates with Titan formation, and secondly, whether you've done a broader analysis of antifungal drug resistance with Titan cells.
Yeah, okay. Frustratingly, Titanization does not correlate with fluconazole resistance. It seems like it should, right? But the major conclusion of one of my PhD students over the last 2 years has been that there does not seem to be a direct relationship between the ability of a cell to form a true Titan cell and the ability to resist fluconazole. And we're basing that on a really nice data set from Neil Stone and Judy Berman and Titanides, showing that cells that have come straight from a patient and then have been passaged on fluconazole, so these are the heteroresistant strains, there's no correlation between the ability to become heteroresistant and the ability to form Titan cells in that initial population. However, having said all of that, Elaine also has asked a question about regulation. It's becoming clear that the ability to form a Titan cell is a highly regulated process and is regulated at multiple different levels by many, many different pathways. So just because a cell doesn't make a true Titan cell that's large, polyploid with a distinct capsule and a distinct cell wall, doesn't mean that it doesn't have the regulatory mechanisms to undergo parts of that phenotype, right? So changes in ploidy and ability to tolerate changes in ploidy is probably regulated at a much larger level than simply loss of, or plus or minus this Titan phenotype. What was the second part of the question I was supposed to ask?
So the two questions were the fluconazole resistant and whether you've tested more broadly the
Oh yes, and whether it's resistant. Yeah. Okay, so because what we're observing is changes in ploidy, and that's probably what's driving the drug resistance, there's no reason to think that that is specifically related to fluconazole, right? It's probably any drug resistance phenotype that can be linked to aneuploidy is probably going to also pass, right? So just the ability to become aneuploid allows you to resist stresses because you're able to deal with them in new ways, if that makes sense. We haven't specifically tested different panels of drugs. What we have done is look for chemical inhibitors of the process and shown that if you inhibit titanization, you can then change the ability to resist fluconazole.
Okay, thanks.
Bruce, there's a question whether you were able to isolate and sequence the blastomyces isolates from these patients.
So the isolates, or at least representative isolates from some of the patients have been obtained. They reside in the Marshfield Clinic laboratory. To my knowledge, they have not been sequenced. One of the interesting features of blastomyces in terms of looking at the pathogen as opposed to the host is that two different species of blastomyces circulate in Wisconsin, in fact. Blastomyces dermatitidis and a more recently described species, Blastomyces gilcrestii. There are now probably at least 6 different species of blastomyces, and in our state, we see both species circulating, dermatitidis and gilcrestii. What's interesting about the 2 species that circulate in our state is that in fact they do direct different clinical outcomes. Dermatitidis has a much greater propensity to disseminate outside of the lung as compared to gilcrestii, which is more often restricted to the lung in human patients. So, so while we know there are different outcomes, I can tell you that at least I don't know, and I don't believe that the isolates have been sequenced. My suspicion, but this is a speculation, is that they're probably gilcrestii, but it's worth looking at.
Okay, we have just less than 5 minutes, so we're going to ask our speakers 1 question each to end, and then we'll close the session. So the last question, which is one you've already alluded to, Liz, but I think it's worth just a few more quick comments. It's about the mechanism of positive and negative regulation, and what is known about that, whether it's epigenetic or the mechanism.
Yeah, this is really more than a 1 minute answer.
You got 1 minute. I'm going to time it very carefully.
I mean, this is what we've been working on in my lab for the last, you know, I don't know, 100 years. How many has it been since January? It's clear that there are transcription factors involved. You knock out a wide range of transcription factors, you definitely see phenotypes. And so that's been shown previously by a bunch of work that Kirsten did in the in vivo models. It's also, we found USB101 was a transcription factor. There's a new paper from Tamara Dorings identifying another transcription factor that's involved. So it's clear that there's a transcriptional network that's regulating this process. Probably also post-transcriptional regulation, which is the major focus of my research. So I didn't talk about it at all today, but we have a lot of evidence showing that there are redox regulation of this process. If you stimulate redox mechanisms, you get more Titan cells. If you block redox mechanisms, you get fewer Titan cells. There's sensitivity to redox stress. Is a very clear sort of process involved there. In terms of epigenetic regulation, I'm sure epigenetics influences everything, right? It's sort of the reason that field is so fascinating. And I'm sure that there is some role for example, HDACs and things like that in the process, but we haven't specifically investigated that.
Okay, thank you.
Okay, and there's a lot of really great questions still in the feed, so I apologize to everybody whose questions we did not get to, but we're gonna end with a question I happen to know is from a postdoc, not mine, asking if you see a conserved gene deficiency in the IL-6 gene among susceptible minority populations. And I guess that could apply to blasto, but maybe to other fungal infections as well.
So I'm sorry, I didn't quite understand the question, Sarah.
So the question is, do you see a conserved gene deficiency in the IL-6 gene among susceptible minority populations?
So I guess this awaits further study. I mean, it'd be particularly interesting to look at minority populations that are vulnerable, not just to blasto, but actually to other fungal diseases. As I mentioned in the beginning, you know, for this field, the endemic mycoses, this is really one of the holy grail questions. What accounts for variable susceptibility? For example, in cocci, it's been for the last, I would say, similar to Liz, the last 100 years, this has been a question sort of bouncing around as to why Filipinos are so vulnerable to coccidioidomycosis in getting disseminated infection. And we see in Native Americans that they're vulnerable not just to blasto, but also to cocci. So this is a question, I guess, or this is a finding that begs additional study in these populations. And it would not be surprising to me that there are variations at this particular locus. So to be continued. And since that's the question, I wanted to personally extend a thanks to you, Sarah, and to you, Gordon, for inviting me and inviting us to this really exciting new forum. It's really been fantastic. And I enjoyed watching the first one, and I really enjoyed participating in this one too. So thank you, thank you, thank you.
Well, Bruce and Liz, thank you so much for taking the time to get it. You're both now will be immortalized on YouTube. So if you want to go back and watch your own presentations, you're more than welcome to. I want to thank all the attendees. Thank you so much for joining us today. And thank you for all this fantastic list of questions. I'm very sorry Sarah and I didn't have time to get through them all. I'm sure that you would be welcome to contact Liz and Bruce directly if you have some burning questions that you feel were not covered in the short time that we had. So thank you all once again for joining us, and we will see you in 1 month from now at the next MycoTalks. Thank you and goodbye.
Stay safe.
Stay safe.